Hello everyone, my research is to study the metabolism pathway of a neuropathological signature protein, TDP-43, and my research work has been published in Journal of Cell Science last year. And why TDP-43 protein is so important? Because in 2006, one science paper first found that the hyperphosphorylated, truncated, and polyubiquitinated TDP-43 protein is the major component of aggregates in frontotemporal lobar degeneration and ALS patients. And under normal condition, intracellular proteins are mostly degraded through proteasome and autophage degradation pathway. In addition to proteasome and autophage degradation, TDP-43 is also degraded through CMA degradation pathway. In normal condition, cellular proteins are degraded majorly through proteasome and autophage degradation pathways. In addition to proteasome and autophage metabolism, I first found that TDP-43 proteins is also degraded through chaperone-mediated autophage pathway. Here we can see that TDP-43 protein is co-localized with the lysosome membrane receptor LAMP2A. And within the ubiquitin aggregates found in ALS and FTLDU patients, predominant truncated TDP-43 proteins are discovered, including TDP-35 and TDP-25. And I found that the cleavage of full-length TDP-43 into truncated TDP-35 and 25 fragments is the intermediate step for normal TDP-43 degradation. So, when we compare the protein half-life of the mutant TDP-43 that cannot be cleaved by caspase-3 into truncated TDP, its protein half-life will be prolonged. Within the aggregates of FTLDU and ALS patients, abundant TDP-43 fragments are observed. And here we found that cleavage of full-length TDP-43 into TDP-35 and 25 fragments is the intermediate step for normal TDP-43 protein degradation. And the TDP-43 mutant that cannot be cleaved by caspase-3 into 35 and 25 fragments, its half-life were prolonged. And many proteasome and autophage-related genes are downregulated in ALS and FTLDU patients. And because of the dysfunction of these degradation-related genes, it will cause the accumulation of TDP-43 proteins within cytoplasm. And accumulated TDP-43 and fragment TDP-25 and 35 proteins are prone to form aggregates. Numerous proteasome and autophage-related genes are downregulated in ALS and FTLDU patients. Dysfunction of these degradation-related genes will cause TDP-43 proteins aggregates formation. Numerous proteasome and autophage-related genes are downregulated in ALS and FTLDU patients, and dysfunction of these genes will cause the truncated TDP-43 and full-length TDP-43 to form aggregates. Here we first found that TDP-4325 and 35 fragments play a role as the seed for TDP-43 aggregates. When truncated and full-length TDP-43 reach the threshold of minimal amount, this protein will form aggregates in the cell. So in the metabolism and aggregation pathway of TDP-43, I first found that chaperone-mediated autophagy is also involved in TDP-43 protein metabolism, and the truncated TDP-43 is the intermediate step for TDP-43 protein degradation. So in the metabolism and the aggregation mechanism of TDP-43, we found that chaperone-mediated autophagy is also involved in TDP-43 protein metabolism, and the truncated TDP-43 is the intermediate step for proteins degradation. In the degradation and the aggregation formation mechanism, we first discovered that chaperone-mediated autophagy is also involved in TDP-43 metabolism, and the truncated TDP-43 play a role as the intermediate step for TDP-43 protein degradation. In addition, the truncated TDP-43 is the seed for TDP-43 aggregates formation, and once the full-length TDP-43 reach the threshold, the TDP-43 aggregates will start to form.